The UV-Vis spectroscopy can be carried out by using UV-Vis spectrophotometer. The UV-Vis spectrophotometer uses the visible light and ultraviolet to analyze the chemical structure of the substance. A spectrophotometer is a special type of spectrometer which is used to measure the intensity of light, and the intensity is proportional to the wavelength.
This is ultraviolet visible spectroscopy, which is provided in Vis Lab. As we can see, here is a control board and a sample to place our cuvette. There is 6 cuvettes that can be placed simultaneously, and the test can operate by clicking the button which solution that we want to test. While the test is running, we should close the cover of sample compartment in order to avoid oxidation happens.
In the theory, ultraviolet visible spectroscopy or ultraviolet visible spectrophotometry (UV-Vis) refers to the absorption spectroscopy or reflectance spectroscopy in the ultraviolet visible spectral region. This means it uses light in the visible and adjacent ranges. The absorption or reflectance in the visible range directly affects the perceived color of the chemical involved. In this region of the electromagnetic spectrum, atoms and molecules undergo electronic transitions. Absorption spectroscopy is complementary to fluorescence spectroscopy in that fluorescence deals with transition from the excited state to the ground state, while absorption measures transition from the ground state to the excited state. Ultraviolet and visible radiation interact with matter, which causes electronic transitions, which is in the promotion of electrons from the ground state to a high energy state. The ultraviolet region falls in the range between 190 to 380 nanometer, while for the visible region will fall between 380 to 750 nanometer. As we can see in this following diagram, there is a bonding, non-bonding, and anti-bonding. Bonding, which has sigma and pi, non-bonding has and anti-bonding has pi star and sigma star. And this diagram shows that it is the electronic transitions that are possible. The first one is pi to pi star transitions, and the second one is the n to pi star transitions. Third one is the sigma to the sigma star transitions, and the last one is the n to the sigma star transitions. Take note that the sigma to sigma star transition, it requires an absorption of the photon with a wavelength which does not fall in the UV-Vis range. Thus, only pi to pi star and n to the pi star transitions occur in the UV-Vis region that can be observed. If a particular wavelength of ultraviolet or visible radiation can be isolated from the source and passes through a sample which can absorb some of the radiation, then the transmitted light intensity will less than the incident light intensity. So, the amount of the light intensity with respect to the incident light is called transmittance. The formula for the transmittance is T equals to the transmitted light intensity over incident light intensity. Sample can absorb all or none of the incident light, and therefore transmittance always quoted as a percentage. So the formula of it is the percentage of transmittance equals to the transmitted light intensity over incident light intensity and times with 100%. So from that we can calculate the absorbance. The formula of the absorbance is the A equals to negative log 10T, which is the transmittance. And after we substitute the value of T, we will get the absorbance equals the log 10 times with the incident light intensity over with transmitted light intensity. Okay, so for of percentage of transmittance, which is equal to 0 and 100, the corresponding absorbance values will be 0 and 2 respectively. By plotting absorbance versus wavelength, an absorbance spectrum is generated. Next, there is a tree law. The first one is the Lambert's law, which is the absorbance is proportional to the path length of the absorbing mediums. And the second is the Beer's law, which is the absorbance is the proportional to the concentrations of the sample. And the third law is the combination of the Lambert's and Beer's law, which is known as the Beer-Lambert's law, which is the absorbance is the proportional to the concentration and also the path length. So the formula of this law is the A equals to the ECL. A is the ratio and therefore there has no units. And the constant E is called the molar extinction coefficients, C is the concentrations, L is the path length. So, the Beer-Lambert's law states that the absorbance of the solution is directly proportional to the concentration of the absorbing species in the solution and the path length. Thus, for a fixed path length, UV-Vis spectroscopy can be used to determine the concentration of the absorber in a solution. It is necessary to know how quickly the absorbance changes with the concentration. This can be taken from references, tables of molar extinction coefficient, which is the E, or more accurately can determine from a calibration curve. A UV-V spectrophotometer may be used as the detector for the HPLC. The presence of an analyte gives a response assumed to be proportional to the concentrations. For accurate result, the instrument's response to the analyte in the unknown should be compared with the response to a standard which is very similar to the use of the calibration curve. The response in an example with a peak height for a particular concentration is known as the response factor.
Next, I will explain about the working principle of UV-Vis spectroscopy. Instrumentation and working of the UV spectrometers can be studied simultaneously. Most of the modern UV spectrometers consist of the following parts: light source, monochromator, sample and reference cells, detector, amplifier, and recording devices. The figure shows the components of UV spectrometers. Light source. Tungsten filaments lamps and hydrogen deuterium lamps are most widely used and suitable light source as they cover the whole UV region. Tungsten filament lamps are rich in red radiations. More specifically, they emit the radiations of 375 nanometer, while the intensity of hydrogen deuterium lamps falls below 375 nanometer. For monochromator generally composed of prisms and slits. The most of the spectrophotometers are double beam spectrophotometers. The radiation emitted from the primary source is dispersed with the help of rotating prisms. The various wavelengths of the light source, which are separated by the prism, are then selected by the slits. Such the rotation of the prism results in a series of continuously increasing wavelengths to pass through the slits for recording purpose. The beam selected by the slit is monochromatic and further divided into two beams with the help of another prism. As for the sample and reference cells, one of the two divided beams is passed through the sample solution and second beam is passed through the reference solution. Both sample and reference solution are contained in the cells. These cells are made of either silica or quartz. Glass can be used for the cells as it absorbs light in the UV region. Next, detector. Generally, two photocells serve the purpose of detector in UV spectroscopy. One of the photocell receives the beam from sample cell and second detector receives the beam from the reference. The intensity of the radiation from the reference cell is stronger than the beam of the sample cell. This results in the generation of pulsating or alternating currents in the photocells. Amplifier, the alternating current generated in the photocells is transferred to the amplifier. The amplifier is coupled to a small servometer. Generally, current generated in the photocells is very low intensity. The main purpose of amplifier is to amplify the signals many times so we can get clear and recordable signals. Last but not least, recording devices. Most of the time, amplifier is coupled to a pen recorder which is connected to the computer. Computer stores all the data generated and produces the spectrum of the desired compound.
I'll ignite the source lamp, creating a variety of wavelengths of different light. This light passes through the first slit of the monochromator, ensuring that all of those light photons are traveling along parallel pathways, so that when they strike the prism, they are refracted into a rainbow of colors. So each wavelength of light is moving to a different place in space. So only one wavelength of light in this situation is going to make it through the second slit in my monochromator, striking the beam splitter and becoming two beams of equal intensity. These two beams of equal intensity will traverse a cell, a different one for each of course, one being the reference and one being the sample cell. As the beam exits these cells, it strikes the detectors, which are firing away, creating an electrical current. And you'll notice at the moment that the intensity of the light exiting both the reference and sample cells is identical.
The sample will be placed in the cuvette, which is the container must allow the radiation to pass over the spectral region of interest. The cuvette must be rectangular and the internal width is within 1 cm. The test tube also can be used as the cuvette. The widely applicable cuvette is a fused silica or quartz glass because it is transparent throughout the UV. Journal 1 describes the structural and optical properties of hexagonal zinc oxide nanostructures grown by ultrasonic spray CVD. Zinc oxide materials have been widely used in the optoelectronic applications due to its direct wideband gap and large excitant binding energy at the room temperature. Zinc oxide can be grown with differently shaped nanostructures with hexagonal shape, the flower shape, and also the star shape. From the figure, it shows the transmittance spectrums of the samples. Based on the figure, we can analyze that the optical transmittance of samples are decreased with the increment in ultrasonic transducer power. The zinc oxide nanostructure have an absorption peak at 380 nanometer. Therefore, in the transmittance spectrums of the samples, a radical decrement at approaching 380 nanometer is observed in all samples, while sample C has a very low optical transmittance in the visible area among the samples.
The sample A and B are transparent over the 70% in the visible area. The reduction of the transmittance of sample C might be explained the strong scattering of the light from the surface and due to the rough and the increased thickness of the sample, and thus lower ultrasonic transducer power from 9 volt can be used for the better transparent zinc oxide nanostructures. It is shown that transmittance of the sample C has been decreased at near 500 nanometer of wavelength, which known as the green emissions, because of the recombinations of electrons with holes in ionized oxygen we can see in the sample. In Journal 2, it described the optical, electrical, and photoelectric properties of nanocrystalline cadmium selenide films for photosensor application. The optical properties of the films were studied by using the spectrophotometer JASCO model V570 UV-Vis NIR. The optical band gap of film was determined as a function of the film thickness and showed high band gap compared to bulk material due to quantum size effect, and usually for the wavelength in the range of 200 to 2500 nanometer. Next, as we can see the following graph, which shows that the transmittance and reflectance against the wavelength. So, from that, we can know the value of the transmittance and also the reflectance. Okay, based on the graph, we can see the transmittance of the film decreases with increasing film thickness and the corresponding reflectance increases. In addition, the film have strong absorption in ultraviolet and a wide range of visible light region. In near-infrared region, the film show low absorption and it is strong and low absorption region depends on the film thickness. Besides, the optical band gap of the film was found to decrease from 2.55 to 2.07 electronvolt by increasing film thickness from 101 up to 179 nanometer.